Okay, you need to differentiate meiosis from mitosis. Mitosis occur at somatic cell, which is for growth and replacement of that cell. You need to grow. Okay, example from gametes to from zygotes to embryo and then to fetus, and then we grow up. Okay, and then to replace your skin, your dead cells, and then your hair. That's for mitosis. And the daughter cell is genetically identical to their mother cell. Maksudnya kalau parent cell ada 2N, daughter cell pun akan ada 2N. So genetic material dia sama, exactly a copy, macam kita photostate. But for meiosis, it is specifically designed for gametes, which is the main function is to reduce, ataupun the main purposes of meiosis is to reduce the number of parental cell, parental chromosome set from diploid to haploid. Kita nak tukarkan, kita nak reducekan daripada 2N kepada 1N. Itu fungsi meiosis. It only occurs at gametes, okay? So reproductive cell. So it can occur in plants and also in animal cells. For in plants, it can occurs in spores and pollens and eggs, okay? The ovum, okay? In plants, okay? And then in animal, it can occur in sperm and also ovum. Okay, so meiosis process will produce 4 daughter cells instead of 2, just like mitosis. Kalau mitosis dia hasilkan 2 daughter cell, meiosis it produce 4, okay? 4 daughter cell, and each of the daughter cell is N. Okay, dia dah reduce number daripada 2N kepada N, dan dia ada 4 daughter cells. Okay, and then the genetic content for meiosis is not the same, which means it different from their mother cells. Okay, so genetic content dalam sperm dan ovum tu tak sama dengan yang dalam parent cell lah. Okay. So sets of chromosome in human cell. So human have chromosome sets. Nampak terbalik ya? Macam nak lukis 2N equal to 46. Okay, 2N equal to 46. Okay, and if you want to reduce it, it will become N equals to 23. So this is the purpose of meiosis. Daripada 2N nak menjadi 2N. Okay, so kalau bentuk 2N ni kita panggil dia diploid, and kita panggil dia haploid. Okay, so you need to remember. Okay, the sex chromosome will determine an individual sex. Okay, chromosome sex akan menentukan jantina kita. So for female, it will have XX. Okay, so untuk perempuan kita akan ada kromosom XX, double X. Okay, but for male, it will have X and Y chromosome like this. So this is female, this is for male. Okay, this is for female, this is for male. Okay, okay. XX chromosome ni menentukan jantina perempuan, XY untuk lelaki. Right. So human males have only one X and one Y. Okay, and then kita ada 23 set kan, 23 set, 23 set kromosom, satu set adalah untuk tentukan jantina ataupun kita panggil sex chromosome. Lagi remaining baki 22 lagi, which is kita panggil sebagai autosome. Okay, so one set is for from 23 set of 23 pairs of chromosome, one pairs for the sex chromosome in order to determine either it is for male or female. Okay, and the remaining 23 tolak 1 tinggal lagi 22, and then the remaining 22 pairs of chromosome is called as an autosome. Okay, so the 46 chromosome in a human somatic cell are 2 sets of 23. So yang 46 tadi ni, okay, 2N equal to 46 ni is actually coming from father and mother. Maksudnya sekarang satu set coming from mother plus another one set from father, dia akan jadi 2N lah. That's why ini 23, 1N 23 tambah lagi 23, dia akan jadi 2N equals to 46. Okay, itu datangnya kita lah, atau ni kata sperm from father, ovum from mother, and this is the baby. Okay, the baby is 2N equal to 46. 46 sets of chromosome. Okay, So 46 chromosome in single cell mitosis reveal that there are 2 chromosomes of each 23 types. So it's actually, if you're referring to the slide number 38, okay, in our lecture, there it actually show us the karyotypes of human chromosomes. So human chromosome ataupun kromosom ni sebenarnya dia ada pair dia, kita panggil dia homologous chromosome. So dia susun kalau you refer kepada slide nombor 38 ni, kat sini ada nombor 1, 2, 3 sampailah nombor 22, which is it refer to the autosome, and another one is X and Y chromosome. Itu menunjukkan karyotype ni untuk orang lelaki lah. Okay. And kalau you tengok kromosom set number 1, number 2, number 3, number 4, number 5 ni, dia berbeza, different in terms of sizes. And also dia ada dua, dia macam sepasang. Itu sebenarnya adalah homologous chromosome. Okay. So homologous chromosome dia ada macam ni, dia bukan sendirian, it works in pairs. This is one homologous chromosome. Okay, kalau kromosom saja kita panggil dia macam ni kan. Ni single lah. Ini satu kromosom. Kalau dia macam ni dua, dekat-dekat, dia adalah sama saiz, sama tinggi. It is a homologous chromosome. Okay. Okay, then these homologous chromosomes, okay, refer to the next slide, slide 1039. The 2 chromosomes composing a pair have the same length, centromere position, and both carry genes controlling the same inherited characters. So maksudnya sekarang sepasang homologous chromosome ni, one from the father, one from mother. Maksudnya dia sama bentuk, sama size, and then dia bawa gene yang sama. It carry the same inherited characters. Okay. Sebab kat sini ada banyak loci kan. So loci untuk yang nombor 1 dengan nombor 2 ni adalah untuk genetik ataupun traits yang sama. Contohlah dekat loci nombor 1 ni, belah sini untuk warna anak mata daripada father kan. So yang belah sini, belah satu lagi pasangan ni, tempat yang sama, loci yang lebih kurang kedudukan yang sama, tapi different kromosom sini untuk warna mata juga, tapi karakter tu datang daripada mak. Okay. So sebab dia, and this homologous chromosome carry genes that control the same inherited characters. Cuma mungkin berbeza. Ayah ni warna mata dia coklat, mak ni warna mata dia biru. So anak dia akan dapat mata coklat atau mata biru, we are still not sure. Okay. So dia ada dua traits lah kat situ untuk warna anak mata, for example. Okay. Okay. So if you refer to the slide 39 also, so dalam sel tu, it draw you three chromosome that have blue color but different length. Okay. And another 3 chromosome that have red color, also different length. But if you notice, warna biru ada panjang sama dengan warna merah. Sama juga dengan warna merah. Warna biru sini dia macam kromosomnya dia berdasarkan kepanjangan. Ini panjang kan? Okay, ini sedang ada panjang, ini kecil, for example lah. So dia dah pairkan. So for example, the black chromosome ni is from the paternal set, and this red chromosome is from the maternal set. So kita panggil ini satu homologous chromosome, satu set, dua set, tiga set. Okay. So now we have maternal set of chromosome 3. Okay. Maternal yang warna merah tadi 3, paternal kita ada 3. So kat sini berapa bilangan N dia? 6 kan? 1, 2, 3, 4, 5, 6. So we can write like this. 2N equals to 6. Maaflah tulisannya terbalik kan? 2N equals to 6. Okay. 2N sama dengan 6. Jadi, so apa 2N sama dengan 6? Sebab kita ada 1, 2, 3, 4, 5, 6 kromosom. And then this is a homologous chromosome. One pair from the paternal, another chromosome, one is from paternal, one is another one is from maternal. Dia sepasang lah, satu daripada ayah, satu daripada mak. Okay. Tapi saiz yang ni berbeza dengan saiz yang ni, and saiz yang ni juga berbeza antara yang ni. So dia berbeza saiz dan juga position of the centromere. Okay. But untuk yang set yang ini, dia ada sama saiz, it has same length, centromere, the same centromere position, and they are carrying the same inherited characters. Okay, homologous chromosome. The next slide, okay, homologous chromosome with one chromatid. So contohlah chromosome with one chromatid, nak jadi homologous chromosome tu macam mana So first of all, this is chromosome with one chromatid. When it undergo duplication, it will become like this, okay, that have a sister chromatid. Ini kita panggil sister chromatid. So ni warna biru, warna merah, warna hitam. Kita anggap dia daripada paternal lah, okay. And the other sets, okay, for example, mula-mula dia macam ni So this is a chromosome with one chromatid, and then it duplicated with this one, okay, and this is the maternal set. So bila kita combine ni tu dua-dua, okay, ni kita panggil dia sebagai homologous chromosome. Okay, saya tulis saja untuk pendek lah. So this is actually one from paternal father, one from maternal, which is the mother. This is a homologous chromosome. Okay, so meiosis reduces the number of chromosome sets from diploid to haploid. Yes, we know this. In gametes, especially in sperms or eggs, it is haploid, which is N. For human, the haploid number is 23, N equal to 23. Different species have different number of chromosome. Untuk human, kita ada N sama dengan 23 lah. Okay. And many steps of meiosis 2 resemble steps in mitosis. Of course, step dalam meiosis sama saja macam mitosis, especially meiosis 2. Okay, both are preceded by the replication of chromosome. Okay, however, in meiosis there are two consecutive cell division, which are the meiosis 1 and meiosis 2, which results in 4 daughter cells. And each final daughter cell has only half as many chromosomes as the parent cell. So it means that here the major differences between meiosis is meiosis have two times of division, but in meiosis 2 the step is similar like mitosis. So I will draw the meiosis part here later. Okay. So next we go to the importance of meiosis. So why certain cell need to undergo meiosis? It is important because it is a basis of sexual reproduction. Crossing over during meiosis can produce the variation in the chromosome gametes. So we have a few events that are important during meiosis. One of them is the crossing over, okay, yang berlaku waktu meiosis 1. Okay, so this crossing over will produce the variation of chromosome. Thus, fusions of gametes will increase the variation in the characteristic of individuals. Maksudnya, it will produce variety of characteristics. Okay, kita nak crossing over tu waktu meiosis 1 tu kita nak perbagaikan gametes. Okay, tak adalah semua adik-beradik sama je muka. That's why and then stages in meiosis. Okay, we have two stages. After chromosome duplicates, two division will follow. The first one is meiosis 1. So yang ni kita panggil meiosis 1 sebagai reductional division. So reductional division, what the important event that occur here is homologous chromosome will pair up and separate. So waktu meiosis 1 kita ada homologous chromosome, resulting in two haploid daughter cell with replicated chromosome. Okay, kita ada meiosis 1, kita ada homologous chromosome. Okay, saya lukis kat sini sepasang je lah supaya senang faham. And then after we finish meiosis 1, kita akan ada we will have two haploid daughter cell with replicated chromosome. Kita akan ada dua daughter cell. Ni baru dua, okay. And then when meiosis 2 is finished, so meiosis 2 we call as the equational division, sister chromatic will separate. Okay, so bila meiosis, so this is meiosis 1, meiosis 1 dia akan hasilkan 2 daughter cell, and then proceed with meiosis 2, it will produce another 2 daughter cell, which we will have 4 daughter cell. Okay, I draw it first. So jadi macam ni. So yang ni nampak, yang contohlah this is the black chromosome, okay. And then bila habis meiosis 2 dia akan jadi sister chromatic macam ni, okay. And then this is bahagian another set, jadi merah, dia separate kan. So this is mula-mula meiosis 1 dia akan separatekan homologous chromosome menjadi macam ni. Meiosis 2 dia akan separatekan, it will separate the chromosome into the sister chromatic. Ni kita panggil dia chromatic lah, okay. So habis meiosis 2 akan hasil dia 4N, okay, 4 daughter cell. Bukanlah bilangan N tu jadi 4N, no. This is 2N. After meiosis 1 dah jadi N, and this one also N. N, 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 N. Okay, bilangan dia semua haploid lah ni, okay. Okay, next, okay, stages of meiosis. Division in meiosis 1 is separation of homologous chromosome. Proceed with prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. And then division of meiosis 2, separation of sister chromatic. So you need to remember meiosis 1 untuk separate homologous chromosome. Homologous chromosome tu yang mana? Yang ni, dia sepasang ni, daripada mak satu ayah satu. Ni homologous chromosome. Kita nak separatekan dulu homologous chromosome waktu meiosis 1. Okay, bila dah separate, kita akan proceed dengan meiosis 2. So meiosis 2 dia akan separatekan sister chromatic. Yang ni tadi ni, bentuk butterfly ni dah tak ada, dia bagi 2. Okay, so ni kita panggil dia sister chromatic lah, ataupun chromatic. Okay, proceed with prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis also. Cuma je kat sini, okay, you need to remember interphase berlaku before prophase 1 only. Okay, cuma sebelum prophase 1 saja berlaku interphase. So after finish telophase 1 and cytokinesis, tak ada lagi interphase dah. Dia cuma ada kita panggil interkinesis, macam gap sekejap between meiosis 1 and meiosis 2. Tapi no more interphase, right? Once sekali saja ada interphase tu. Okay, so you referring to the first interphase. Okay, kita tengok slide number 49. Interphase is the resting stage. I think you already know this. Okay, we have discussed the interphase in detail previously. Okay, this is the preparation stage for meiosis 1 and meiosis 2. And chromosome duplicated during S phase in interphase before meiosis 1. Okay, and meiosis 2. Okay, so the prophase 1. Okay, we are going to the first step of mitosis, which is prophase 1. Okay, so in prophase 1 we have event, which is an important event that occur here. Okay, so prophase 1, what happened is, okay, during prophase 1 you need to understand what happened for each stage. Sama juga macam mitosis kan, you kena tahu prophase apa yang berlaku. Sama juga macam meiosis pun, you need to know what happened during prophase 1. First of all, chromosome begins to condense. Okay, daripada chromatin structure dan interphase tadi dia start condense, and then the nuclear envelope and nucleolus will disappear and spindle will form. Centrosome migrates to opposite poles of cell. So you tengok gambar dekat dalam slide 50 tu, okay, dia dah pair up the homologous chromosome. Okay, this homologous chromosome begin to pair up in a process called synapsis. So synapsis tu adalah dia nak pasangkan homologous chromosome. Okay, so kalau kita ada 3 pasang homologous chromosome, okay, so draw lah 3 but different sizes of the chromosome lah. So the process of pairing up this homologous chromosome, we call that as a synapsis. Okay, synapsis. This is the first event that occur during prophase 1. So when the homologous chromosome are already paired up together like this, This structure also known as the bivalence. So bentuk yang macam ni, dia dah pasang-pasang ni, kita juga panggil dia sebagai bivalent ataupun tetrads. Okay, bivalent. Alright. And then in crossing over, okay, after synapsis akan berlakulah crossing over, which is non-sister chromatid will exchange DNA fragment. Apakah yang dimasukkan dengan non-sister chromatid? Okay, so tadi contohlah saya zoom out this one pair. Okay, kita keluarkan besar, lukis besar kat sini. Satu from paternal, another one from maternal. So kalau yang ini dua sister chromatid, okay, yang ini dua pun sister chromatid lah sebab dia dalam satu kromosom. But if we took this side satu, belah sini contohlah kita bagi dua, okay, and this side satu lagi, this is non-sister chromatid. Kenapa? Sebab dia bukan daripada satu kromosom yang sama, dia berlainan. Satu belah sini dengan satu lagi yang daripada sini yang hitam ni, ni kita panggil non-sister chromatid. So waktu crossing over, non-sister chromatid ni dia akan exchange DNA segments. Dia akan exchange at a place berlakunya chiasmata. Okay, chiasmata which is the X-shaped region where crossing over occur. So mula dia macam ni kan. So waktu chiasmata, saya kira ni, waktu chiasmata dia jadi line sikit. Dia macam bertukar satu sedikit region kat situ. Saya lukis. Saya tak pandai sangat lukis tapi lebih kurang lah ya. Macam ni. Sepatutnya yang sini warna hitam kan, semua kromosom ni maternal ni. Okay, but now it's exchanged a little bit here. It takes up the red DNA supaya masuk dalam sini. And then it exchange with the red chromosome ni, DNA dia warna hitam. So bila dia dibentuk macam ni, kita panggil this is a chiasmata. C-H-I-A-S-M-A-T-A. So we call this structure when exchanging DNA material ni chiasmata, a region where crossing over occur. So for semua you kena remember, satu, synapsis, which is the pairing up of homologous chromosome. Okay. Second, you need to know is crossing over. So crossing over berlaku antara non-sister chromatid of the homologous chromosome. Where did the crossing over occur at the non-sister chromatid? We call that as at the chiasmata. Okay. Chiasmata is the X shape like this, X shape, okay, region where the crossing over occur. So it can occur at this side, or it can occur at upper side only, or it can occur both simultaneously. Okay, dia boleh berlaku belah bawah saja, ataupun atas saja, ataupun boleh berlaku atas dengan bawah. Ikut suka dia. Okay. Alright, next in metaphase 1. So now kita dah ada, we have this homologous chromosome yang dah bertukar DNA segments, tapi bukan semua DNA lah, separuh saja, sedikit saja kat bahagian bawah ni at the chiasmata. And then for metaphase 1, it will proceed of the lining up of the homologous chromosome at the equatorial plate, ataupun kita panggil dia metaphase plate. So homologous chromosome tadi yang dah bertukar tadi tu akan beratur dekat metaphase plate. Macam mana dia beratur? So kalau kat sini centriole, okay, sini satu centriole, it will pair up, it will line up here lah. So remember to draw this structure properly. Cara dia susun tu, okay. You need to draw dia baring horizontal lah. Kalau central dekat atas dan kat sini. Kalau central you lukis kat sini, you boleh lukis this vertically, menegak. So now I'm drawing this centriole dekat atas dan dekat bawah. So you need to draw this chromosome secara horizontal. Okay. So if you notice, I susun dia. Okay, merah satu bawah, satu atas, satu bawah kan? Okay. So now tak semestinya susunan dia macam ni. It's not necessarily to do this arrangement. No, it is we call as the independent assortment. It can also line up all black at the upper, all red chromosome below. Ikut suka dia je. Kita panggil dia independent assortment. Okay. So the centromere of each pair of homologous becomes attached to the spindle, one above the other below the equator. Okay. Maksudnya centromere belah atas ni dekat spindle fiber lah, yang belah bawah ni satu dekat bawah. Okay. Both chromosomes still attached to another at chiasmata. So at this phase masih lagi melekat antara lain lah. Maksudnya homologous chromosome masih lagi dalam bentuk homologous chromosome still attached to another at chiasmata. Sebab ada chiasmata tu, dia tak yang tadi tu chiasmata, which is this one ni, dia masih lagi dalam bentuk macam ni lah. And then dia beratur dalam chiasmata tu dekat metaphase plate ni. Okay. So the important event that occur during metaphase 1 is chromosomes line up by homologous pairs. Okay. Now, dekat slide ni dia tak mention about the independent assortment. So sebenarnya ada satu event yang penting yang berlaku waktu metaphase, which is independent assortment. So you tengok dalam modul you, muka surat berapa? Surat 104, 104. It said about the independent assortment. So ini berlaku waktu metaphase 1. Okay, sahaja metaphase 1 sahaja yang berlaku independent assortment ni. So you need to carefully read the sentence here. What does it mean by independent assortment? So, yang saya lukiskan tadi tu lah, yang saya cerita dia tu. So bayangkan ni tak nampak sangat Sebenarnya ni satu line yang warna hitam, ini satu line yang warna kelabu sikit. Line apa ni? Line kromosom lah. Ini boleh jadi possibility of arrangement of chromosome yang pertama. Then it can be like this. Selang-seli pun boleh, ataupun macam ni, ataupun macam ni. So we don't know which pattern yang kromosom tu akan ikut. No, we don't know because it is independent assortment. Dia bebas, dia nak pilih nak susun macam ni ke, macam ni ke, macam ni ke, macam ni. No, it depend on the cell itself. Okay. Okay, so for prophase 1 kita ada event which is crossing over chiasmata and synapses. Okay, event dia adalah crossing over lah. Synapses is when the homologous chromosome to pair up together, and chiasmata is the where the genetic material is exchanged. That is for prophase 1. But when we proceed to metaphase 1, the homologous chromosome line up at the metaphase plate, and then the way they are arranging themselves at the metaphase plate is by independent assortment. Okay, it is independent. Okay, thus tak semestinya setiap kali dia melakukan meiosis dia akan susun paternal kromosom belah atas, maternal belah bawah. Tak semestinya. It is independent. Tak bergantung kepada siapa-siapa pun. Okay. So next move to the anaphase 1. So for anaphase 1, these pairs of homologous chromosome. So tadi yang beratur ni adalah homologous chromosome kan. So kita nak separatekan this homologous chromosome. So this homologous chromosome will be separated. Okay. So you refer to the slide number 52. Okay, sister chromatids will remain attached to one another. No centromere division, only the homologous chromosome have become separated. So maksudnya sekarang kejap So katakan ini adalah homologous chromosome pair yang tadi. So anaphase 1 kita nak separate kan. Okay, ini naik ke atas. Okay, satu pair lagi turun ke bawah. Sama juga untuk yang ini pun, yang ini pun. Okay. So So ni anaphase 1. So anaphase 1 basically is to separate the homologous chromosome. Kita nak pisahkan homologous chromosome menjadi kromosom macam ni lah, satu-satu. Okay. So dia akan jadi satu pol kat sini ada 3 kromosom. Okay. Satu merah, 2 hitam kan. Itu kalau ikut arrangement selalu kes lah. Bagi anaphase 1 dia akan jadi like this. Nanti kita akan pisahkan dulu kat sini lah waktu telophase tu kan. Okay, so this is for anaphase 1. Okay, we want to separate the homologous chromosome. Kita nak separatekan homologous chromosome. Okay, so then proceed to telophase 1 and cytokinesis. So when the homologous chromosome have separated, okay, had been separated, chromosome will reach the opposite poles and then the nuclear membrane and nucleolus will reappear. Okay, cytokinesis will take place before cells enter meiosis 2. And as usual, in animal cell it will form cleavage furrow, and in plant cell it will form cell plate. And then take note that during telophase 1 and cytokinesis, no chromosome replication will occur between the end of meiosis 1 and the beginning of meiosis 2, because the chromosomes are already replicated before meiosis 1. So maksudnya sekarang daripada meiosis 1 nak pergi ke meiosis 2 dah tak ada berlaku interface. No more interface. Kerana waktu interface ni sepatutnya berlaku apa? DNA replication kan. So interface dah berlaku sebelum meiosis 1 ni. So kita tak perlu nak melakukan lagi satu interface 1. Kita tak perlu untuk melakukan sekali lagi interface sebelum masuk meiosis 2, tak perlu. Okay. Dia cuma ada berlaku event yang kita panggil berehat seketika tu, interkinesis if I'm not mistaken. Ni sekejap cari je mana interkinesis tu. So when it comes to itu sajalah. Okay.